IgG has a longer half-life than any of the other antibodies is that the monocytes can grab these receptors and hang on to it. It doesn't go on to degradation and then release it at another time. So it definitely really extends the half-life of the antibody. So in that sense, right, IgG can do a lot of different things. It can get out into the tissues, and it can also go across the placenta. And I just put this in here because this shows is a good example, right, of the role of these neonatal Ig receptors. And what you're looking at here is a newborn, and the red is the antibody. So colostrum has high concentrations. Colostrum is the milk that is accumulated, right, right before parturition. So I work with livestock, right? So you can see the mammary, especially in a young animal, you see the mammary gland expand massively. All the accumulation of all of this antibody, it's a huge amount of antibody. And so the newborn ingests this, and during the first about 48 hours, all of that antibody gets transported by those receptors out of the gut and into the calf. And so you see this massive distribution of antibodies. After that gut closes, you don't see that occurring anymore. Calf's on its own, okay? Those maternal antibodies that they get are going to probably last them quite a while. We know that because of the interference with vaccinations in early life of calves. But it is going to provide intestinal immunity. So they continue to take in antibodies out of the milk, right? It doesn't have it's not highly concentrated like it was in colostrum, but it's still being produced. It's still very efficient at protecting, but only protecting the gut. You're not going to get it moved across into the circulation of that animal. And the reason, I guess the other point I would make about this is when that maternal antibody gets actively transported across, you think, well, that's got a half-life, right? And so it does have a half-life, but you can find that if the dam had a really high antibody titer, say to a certain viral infection that it's experienced over its life, that if you try to vaccinate this animal, say within 2 months of birth, you get no response because there's still so much antibody that calf got from the dam that it neutralizes, if we're talking virus, right? That antibody, that neutralizing antibody is going to neutralize grab all of the antigens that's in that vaccine and not make them available for production of an immune response. So it's a big problem. We see this, but you see the same problems in human medicine. You have all these kind of timetables, right, of when you can vaccinate neonates, and that's because there can be interference from the maternal antibody there. But again, that is because we have the capacity to transport that antibody into the blood. So innate and adaptive immunity working in concert here, okay? So antibody plus complement, right? And so then we need to think of the two classes of antibody that can activate the complement cascade. One being IgM is incredibly efficient, right? It has this planar form. It can't bind complement. It binds antigen on the surface of the microbe. It assumes a staple conformation, and it can bind C1q and activate the complement cascade. One molecule of IgM. If we're talking about IgG, here it's binding to the microbial surface here, but you have to have IgG bound at the density that will put two molecules of IgG very close together, so because it is required to bind C1q. Otherwise, you're not going to activate the complement cascade. So early on in an infection, you probably don't have the enough IgM to create this dense binding that will facilitate the activation of the complement cascade. And that's why IgM is so important, because it takes one molecule, every molecule, single molecule that binds and assumes a staple form, in fact, can activate the complement cascade. So it works out well, right, that IgM is the first antibody that is produced. And again, just to remind you, right, the complement cascade, IgM, IgG, bind C2, you activate this cascade, you get this C3 convertase here, you get all of this C3b produced, right, that's going to opsonize the pathogen. And we already saw the importance of C3b when it comes to activating the B cell as well, right? Because that's going to facilitate when the B cell binds that via complement receptor for this, will facilitate the production of C3d, which is so central in activating that B cell co-receptor we talked about. So you get a lot of C3a produced. They don't even see the products of C5, right? C5a, both of those are pro-inflammatory mediators. They're both chemotactic for neutrophils. And the C5b is going to opsonize that surface as well. And then you can actually get, right, the membrane attack complex formed. And maybe if you're lucky, you'll get some lysis of those bacterial cells. Okay, so what's this? This is what we're looking at here is clearance of immune complexes. And we talked about this when we talked about complement, right? Small antigen-antibody complexes form in the circulation. These are bad news. These are bad news complexes if they get stuck on vessel walls, okay? So what you want to do is you want to eliminate these small immune complexes. And it's just soluble antigen being complexed by antibody. You're going to activate, these complexes are going to activate the complement cascade. And red cells, the red blood cells, have CR1 receptors, complement receptor number 1. That's what binds the C3b. That's what the B cell has on it as well, right? So red cells have type 1 complement receptors on them, and they carry these immune complexes. They collect them. They're like a sponge, okay? And they'll travel through the spleen and the liver, and there phagocytic cells will remove the immune complexes from the erythrocyte surface, and the red cells go on and carry on. So one thing you do not want is a genetic defect in the complement receptor type 1, because all of these things that we've talked about that is so important. And it kind of also goes to show, right, that you can find these cell surface receptors in a lot of strange places. And whoever figured that you'd have type 1 complement receptor on red cells. Okay, so, and this is one of the more obvious slides. We've talked about this. FC receptors, right, for the various heavy chains. Free immunoglobulin does not cross-link FC receptors, okay? And when we say cross-link FC receptors, what I'm talking about, right, is physically creating a link between two cell surface receptors. So it's the same as IG, right? Cross-linking surface IG. It means hooking two of them together with an antigen with some sort of a bridge. And so that's what antibody here on the surface of this bacteria can facilitate that. So aggregation of immunoglobulin on bacterial surface allows the cross-linking of these FC receptors. And that results in activation of the macrophage, leading to phagocytosis and destruction of the bacteria. So here's a antibody that's already coated the bacteria, right? And it has two opsonins on it now. It's got C3b and it's got antibody on the surface. Complement receptor 1 is going to bind the C3b. The FC receptors are going to bind the antibody on that surface. Facilitates the phagocytosis of this. You're going to get signal transduction, and you're going to get activation of this cell. If this is a macrophage, you're going to get the lysosomes fused with the phagolysosome and all that nasty stuff produced in there that we talked about the first part of the quarter. Okay, so blocking colonization of bacterial pathogens on mucosal surfaces, it's huge, right? So colonization of the cell surface by bacteria which bind to the surface via bacterial adhesion. So bacteria have these things called adhesion proteins on their surface that facilitate their binding to mucosal surfaces. Some bacteria become internalized and can actually propagate in these vesicles, okay, because they've internalized themselves into those mucosal epithelial cells. Antibodies against adhesions block colonization and uptake. So it's merely, if you will, it's kind of like a virus neutralization, right? If that bacteria can't adhere to the mucosal surface via these adhesions, if you have antibodies to these, then it's going to move right on through. So if you've got IgA out on the surface that can bind these bacterial adhesions, and that bacteria is just going to get swept right on through and not colonize. So the problem is is this is great in an, if you will, in an immune response. But again, you need to